While waiting for everyone to join, I'll mention, we're really glad to have everyone, especially given this is the last PHIND seminar of the seminar series for this year, but we'll be resuming again, of course, next year, and hope you'll be able to join us then as well. We also wanted to mention that we will be having a new seed opportunity that will launch in early January, so we'll keep an eye out for that, and we'll be releasing details early next year. For our upcoming seminars next year, we have Anthony and Christopher will be presenting on plant-based meats and looking at what that means in terms of immune biomarkers, as well as one of our PHIND fellows, Hannah, will be giving us a talk on her high-resolution MRI work as well. And our speaker today is Seraina, and I will switch it over to Dr. Gary Gold to give her the introduction.
Thank you, Ryan. Well, welcome everybody to the final MIPS seminar of 2021, sorry, PHIND seminar of 2021. We're delighted to have Seraina Duhl, who's a postdoctoral scholar in the Department of Radiology here, who will speak to us today about sensor systems to predict, prevent, and cure cardiovascular disease. Seraina works in the Department of Radiology and also the Department of Mechanical Engineering here at Stanford. She's working on the design of soft robotic medical devices using cardiac MRI with Professor Daniel Ennis in close collaboration with Professor Alison Marsden. Seraina trained as a mechanical engineer at the Federal Institute of Technology in Zurich, Switzerland, obtaining a bachelor and master's degree in biomedical engineering, robotics and control, and earned her PhD in the product development group, focusing on sensor systems for cardiovascular applications in the Zurich Heart Project. She conducted cutting-edge research in implantable sensor technologies in collaboration with the Voris Group at ETH Zurich, the German Heart Center in Berlin, and the St. Vincent's Hospital in Sydney, Australia. She's authored many publications in prestigious journals across the fields of medical devices, artificial organs, signal processing, and MRI. And inspired by engineering, she aims to enhance human health. And Seraina just accepted a position as assistant professor in the Department of Biomedical Engineering and Health Systems at KTH Royal Institute of Technology in Stockholm. So welcome, Seraina, and we really look forward to your talk.
Thank you, Gary, for this really warm introduction, and thank you everybody for joining today. 1% of babies are born with a congenital heart defects. 37% of US citizens will eventually suffer from cardiovascular disease and die from it. 3% of people worldwide suffer from heart failure. And in heart failure, 46% of patients do not survive the first 5 years. Cardiovascular disease costs society 210 billion euros every single year. While these numbers seem quite depressing, we have also witnessed immense progress on digital technologies that give us more and more data on patients' health status. 98% of microprocessors are networked such that they can communicate information with each other. We can now perform 10 million instructions using just 1 milliwatt. And to sequence the human genome, we now only use 1,000 euros compared to 10 million only in 2000. So I see the intersection of these two areas as something where we can learn a lot and invest a lot and get a lot out of it. And I think the German government agreed agrees with this by committing 3 billion to the digitization of healthcare just last year. So to give you a little bit of perspective, historical perspective, I would like to start with talking about the history of medicine, which set us up for the digital health. In around the last 20 years, we have moved from heuristic-based medical treatments into the area of evidence-based medicine, which basically uses data coming from clinical trials and mostly in randomized fashion to inform medical guidelines that are agreed upon on mostly an international level. So these medical guidelines inform treatments that clinicians provide for our patients. Just last year, again, as in the example of Germany, the German government has invested to now finally introduce the digital patient record. In my opinion, the patient record is a crucial step towards connecting medical patient information with smart medical systems that we are all invested in. Already now, information is collected from smartwatches, bedside monitors, implanted sensors, and mobile phones. The only thing that we're missing is really connecting this information with patient data. In a recent study, it was shown that using implantable pressure sensors, hospitalizations in heart failure could be reduced by 1/3, which is a really significant improvement in terms of treatment. I see 3 complementary areas in which technologies developed in engineering can impact patient treatment: prediction, prevention, and cure. And some of you might recognize this from the slogan of Stanford Medicine. I would love if this talk inspires some of you to reach out across interdisciplinary barriers and work together in a translational way. So prediction, prevention, and cure uses data acquired along the time course of a patient with cardiovascular disease. Usually a patient is admitted to hospital for the first time with something like a heart attack. 90% of patients nowadays, because we have improved our treatment so much, survive a heart attack. However, the heart attack also leaves the heart weakened by this event, which is why we want them to be able to predict the onset of more chronic conditions, such as heart failure. Once the patient is diagnosed with heart failure, hospitalizations become more frequent. And that's really where we would want to prevent these hospitalizations to happen again and again, because this is really also what increases the cost of our healthcare system. At a critical point there, we come to this point where pharmaceutical therapies, unfortunately, do not work anymore as well. And then the path diverts, and the only thing that these patients can receive in terms of therapy now are either a transplantation or the implantation of a ventricular assist device. I will talk a little bit more about these devices at a later stage, but just to give you a first idea, if we want to cure heart failure with the implantation of the device, it is really essential that those devices interact in a physiologic way with our patient. My background, as Gary mentioned, is in control theory and has taught me really the importance of sensors. With any time we want to have the environment interact with data. I therefore like to see patient treatment from a control systems perspective as well, where the goal is to maximize health of each person. So a specific person might experience that something has changed in their health status and seek advice of a doctor. At a clinic, there's going to be a lot of sensors and tests that are going to be done, enriching the information that we have about this specific patient. We can measure health and then compare it to a healthy reference, which is usually established through larger patient cohorts. So we can compare this health status to the population health status. If there is a discrepancy between this healthy reference and our patient, we then need to take actions. And the person or the device that takes these actions might be different in every single case. The way that healthcare is set up right now, it's typically the clinician who then administers a therapy. But we can also imagine a situation where the patient is actually encouraged to change their lifestyle, so to take the action to change the lifestyle to get back towards that healthy reference. And as I mentioned briefly before, we now also have these medical devices that might have to be adapted in terms of their operating condition, such that they better match the patient and bring them back to this healthy reference. So in all those cases, it's great if the patient has a perception of their health status. But if we want to increase the timeliness of this loop and of treatment to keep cost and also the incidence of chronic diseases low, is really by replacing this perception by an automated system or by a sensor. So my research really supports the digital health initiative by developing sensor systems and signal processing technologies. I chose this area because it really is the sensors that connect our patients with data. I will now give a short overview over some of the areas that I've worked in. So I have prepared 4 kind of short stories showing going from a clinical problem that is somehow related to heart failure to how we can address this problem using sensor or imaging technology. Before we dive into this, I would just like to share a little bit more about the background of heart failure. So heart failure is a chronic progressive condition in which the heart muscle is unable to ensure blood flow to meet the body's need for blood and oxygen. So in contrast to, for example, an arrhythmic event, which we can treat with a pacemaker, heart failure is really the inability of the heart muscle to eject enough blood in every beat. Treatment options for heart failure, as I mentioned briefly before, are very scarce as soon as pharmaceutical therapies are no longer sufficient. So most of these patients actually should get a heart transplantation, but for some of them, there are no hearts available, such that a transplantation might be delayed. So until now, most of these ventricular assist devices are also called VADs, just help to bridge patients into a situation where they can be transplanted. So after transplantation, these patients have a pretty independent lifestyle again. However, there are 40% of patients that really live for the rest of their lives with such a device. So left ventricular assist devices are implanted in the left side of the heart, which for all those who don't work in the cardiac space is depicted here on the right. So from the left heart, usually the blood flow goes into the left heart and into our cardiovascular system. However, if we have a VAD implanted, there is a second parallel blood flow that provides additional support with blood to the periphery. So the VADs are really implanted in parallel to the native heart. The operating principle of those pumps is that there's a little rotor inside of them that turns at a certain pump speed. And that pump speed is determined once and for all in the clinic by a clinician, just at whatever state the patient is. So if the pump speed is set too low, then the volume of the heart actually it remains pretty increased, which can lead to pulmonary congestion. And on the other side, if the pump speed is set too high, we can affect the contraction properties of the heart. And also we will observe that the volume inside of the heart is decreased. And this is especially problematic also because these pumps are not designed for low speeds, such that the blood that travels through the pump is actually impacted more. So who gets a VAD? Most people who get those implants are adults. A large majority of them are male, where 21% are female. And then there's a couple of children, as I mentioned before, that suffer usually from congenital heart defects that are also implanted with a VAD. However, those look a little bit different. So let's get into the first example. I've talked before about the topic of prediction. So usually we like to predict the onset of critical conditions. But as a mechanical engineer, I also like to predict the way that medical devices at different operating speeds might interact with our patient. So this first story is going to be about that. Outcomes of VAD therapy are worse in females. So females have an increased risk of early 3 months mortality, and they also have an increased risk of right ventricular failure. The underlying mechanisms remain mostly unclear. And at the same time, we have seen these advancements in 3D printing and sensor technology that I've worked on during my PhD. So sharing my work with a good cardiologist friend of mine, Aditi Nayak, we were wondering what the role was of heart size in this discrepancy between outcomes in male and female hearts. And if we look at this a little bit closer, we find that the recommended minimal heart size for VAD implantation is actually not including most women. So most women, because of their heart size, are not eligible for VAD implantation. Now, what we also have to wonder is if for those patients with smaller heart sizes, is there still kind of a problem with those? And our hypothesis was that the current pump speed, so the rotary speed of our pumps, might not always be adequately set for these smaller hearts. So what we looked at in particular was the effect of pump support on the interventricular septum position. So the septum is the wall between the left and the right chamber. And if we start supporting the heart with an LVAD, then we remove effectively blood from the left side and bring it back into the right, such that there is a volume change between the two chambers that might potentially lead to a flipping of the septal wall. So in order to investigate this hypothesis, we used a patient-specific benchtop testing of the septal position. So using 3D printing technologies and a lost wax casting technique, we manufactured these highly detailed heart phantoms. So we had 2 large heart phantoms and 2 small heart phantoms. And then by integrating an ultrasonic sensor, we were able to measure the entire septum from kind of the inside of the heart. And then at the same time, we could change the volumes in the left and the right side, such that we could artificially create this volume shift from the left side to the right side, at the same time observing the position of the septum. At the same time, we also looked at the clinical side of things. So we investigated, Dr. Nayak performed a statistical analysis of a cohort of 15,000 patients and found that heart size explains 14% of the increased 3-month mortality in women and 13% of right ventricular failure. On the other hand side, our benchtop experiments show that in the large hearts at a volume displacement of about 40 milliliters, we do not see a septum shift yet. So that's the middle column. However, in the smaller hearts, at the same volume displacement, the septum already showed a displacement. So in summary, we addressed the problem of worse post-VAD clinical outcomes in women and investigated the hypothesis of if heart size played a role. We then used ultrasound measurements in patient-specific benchtop setting to measure the septum shift at different volume displacements. And in conclusion, we can say that there might be a discrepancy between where we set the pump speed in current VAD therapy, and we encourage people to take this into account when implanting these pumps in smaller sized people. The second story is going to be about prevention. So preventing a disease from progression requires a combination of assessment of the current health status in a timely manner, as well as adequate therapies. The assessment of right heart health is chronically difficult because of the thin chamber walls. And I looked at novel signal processing technologies that could improve the assessment of the right heart in this specific cohort. So the cohort in which we looked at this was Duchenne muscular dystrophy. So these are children that are born with a genetic defects, which happens in about 1 in 5,000 live births. The gen is located on the X chromosome, such that it affects boys only. The genetic mutation causes muscular degeneration in various parts of the body. So these, what I'm showing here, are histological images in mice where this disease was induced. On the patient level, however, the skeletal muscle is typically degenerates first. And then the respiratory system is affected, and lastly, the heart. So the goal of this research was really to find early and sensitive biomarkers to detect the onset of cardiac failure, because that really drives the initiation of medical therapy in these boys. So we have seen previously that the role of cardiac magnetic resonance in those boys is really substantial. First of all, MRI can help us to measure left ventricular volume, so the dilation of the heart, which typically is rather a late finding. The second thing we can do is we can look at focal fibrosis, which are just very densely packed changes in muscular composition and that indicate areas of degeneration. Again, this is also a rather late finding. So the group of Professor Ennis has looked into pre-contrast T1 mapping in the left side of the heart, so to measure diffuse fibrosis, which is actually also elevated in these boys and potentially could precede the occurrence of LGE imaging. So the objective of this study was to characterize differences in right ventricular function and myocardial structure in boys with DMD compared to healthy controls in order to understand if the right heart is as affected as the left heart. So boys with DMD and healthy controls were included in this prospective study and were imaged at 3T at a Skyra scanner. We used BSSFP for standard functional analysis and the MOLDI sequence to acquire the native T1 maps. And then we used a novel way to define the region of interest in these boys. So instead of, as typically done, drawing a circle in the myocardium, we instead draw a single pixel line across the circumference on the RV. And this is important because it might help us to exclude the effect of epicardial fat and the blood mass in measuring those pixels. So in terms of demographics, we had an age-matched cohort. We found that BMI was slightly lower in controls and higher in DMD. And we also found that DMD generally had a higher heart rate. Left ventricular ejection fraction was mostly decreased in LGE positive, so patients that already had some focal fibrosis. And we also found that right ventricular dimensions were rather lower, while also right ventricular mass was lower in patients with DMD. So as we were introducing a novel signal processing method for pre-contrast analysis of RVT1, we first had to validate and make sure that the repeatability of this methodology was adequate. So if we compared just standard measurements in the LV with our measurements in the RV, we find that we were able to include 65 pixels on average in our RV analysis. And in 75% of cases, we were able to do the RV analysis, even though the current state of mind is actually that it's not possible in any case, in children especially. We further found that T1 times in the RV are generally elevated in contrast to the LV, which is in accordance with previous studies in adults. And we also found that even though we were very careful to not include any surrounding blood or epicardial fat, we did have a higher coefficient of variation in terms of the T1 mapping values. If we look at repeatability between scans, between observers, and also within one observer, we did find really good repeatability in the DMD subjects. So using this new signal processing technology, we did see that there was a highly significant increase in pre-contrast RVT1 in DMD compared to healthy controls. At the same time, we also found that similarly to the correlation between LVT1 and left ventricular ejection fraction, we also see a correlation between the right ventricular ejection fraction and right ventricular myocardial properties measured in terms of T1. So there does seem to be a relationship between the structure and the function in both the LV and as well the RV. So we developed a novel signal processing tool in right ventricular failure that addresses the problem of the clinical management of cardiac failure in boys with Duchenne muscular dystrophy. We used non-invasive MRI biomarkers to try to get a better understanding of how the disease progresses in the right heart. And as a conclusion, we found that fibrosis in the RV, based on our results, is likely and should also inform clinical management, which becomes especially important if we think about those patients potentially be supported by a ventricle assist device, which would further strain the right heart. So the third short story, and probably both of the next two actually, have to do with the measurement of left ventricular volume. So an effective way or a reliable way to detect if the heart has trouble pumping is to detect sudden changes in cardiac volume. So during my PhD still at ETH, I invented two types of real-time cardiac volume sensors for two a little bit different applications. So the problem is that current methods to measure left ventricular volume continuously are really very limited. We have external systems that are typically bedside ultrasound, where a clinician has to move with the device to the patient bed every time we perform a measurement. And of course, the gold standard, which is still considered MRI. However, there we even have to move the entire patient towards an MRI system. On the continuous side, we have more implantable systems. The most common hemodynamic measurement tool is the now FDA-approved CardioMEMS St. Jude pressure sensor. However, as you can imagine, pressure and volume still are only related through tissue properties. Then there has been some very preliminary work on attaching sensors to the outside of the heart. However, the materials used in these processes are typically not biocompatible. And then some really important work by Sisk et al. In integrated electric impedance measurements on the cannula of a ventricular assist device. So this is kind of the state of the art of continuous volume measurement. So there really is nothing that we can use on a continuous basis right now. The first example is motivated by cardiac surgery. So cardiac surgery cures a variety of cardiovascular diseases and has really revolutionized the treatment of cardiovascular disease. At the current time, there are 500,000 open heart surgeries performed in the US every year. Each of these patients, however, has a risk of leakage of blood into the periphery, so outside of the active circulation, which then leaves less blood in the heart to make it function effectively. So if we can detect this volume leakage into the periphery early, we can administer fluid and compensate for this effect. This shows that volume monitoring is crucial for post-operative management of anybody really who gets a cardiac surgical intervention. So this is why I engaged in a collaboration with the Biosensors Lab at the Borish Group at ETH Zurich. And we came up with a technology that is soft, so has very similar material properties as the heart, includes biocompatible gold nanowires, and was then manually sutured to the outside of the left heart. So the idea really is that upon changes of volume inside of the heart, the sensor is also stretched more or less depending on the volume inside of the heart. So here you can see the gold nanowires that are integrated in this very soft silicon material. We then implanted the sensor in vivo and exposed the animals to hemodynamic perturbations in terms of infusing volume into the animal to simulate a volume increase or decrease, and also look at the possibility of sudden blood loss and change of the conductive properties in the blood. So what we were able to show is that the strain sensor is surprisingly good at measuring left ventricular volume. So on the left-hand side, you see our gold standard measurements of echo in green, electric impedance measurement in blue, and the volume estimated by our Epi-cardial strain sensor. And you can see that across the entire experiment, we follow those values pretty nicely. On the right-hand side, really what I would like to showcase is just that the variability in terms of volume measurements based on echo shown in green is actually pretty similar to the one that we had based on our strain sensor. So this is really encouraging. At the same time, we do need to say that the strain sensor obviously would need patient-specific calibration of volume, whereas with echo, we really have an effective measurement of volume. The second thing we looked at was really the dynamic response to volume changes. So let's imagine we suddenly remove a lot of volume from the cardiovascular system, which we've done in this experiment, leading to this decline. And then what we did to change the conductance properties of the blood is we reinfused a saline solution. On the left-hand side, you can see how the infusion of saline actually increases the values that we measure with electric impedance. So any electric impedance measurement of volume that we would be using would be affected by the conductance of blood. At the same time, if we combine or if we just use a strain sensor, so a mechanical sensor, obviously we are inert to changes of blood conductivity in measuring left ventricular volume. And the last thing that I would like to show here is something that we use a lot in assessing cardiac work, which are so-called PV loops. So we plot the pressure in the heart versus the volume in the heart. And then by looking at the inside of that cycle, we can show how much work the heart is doing at a certain point in time. So you can say how we can really capture those different hemodynamic states in a similar way as we do with electric impedance in blue. And we can do this as well in strain. And what we also see is that we have less effect of breathing that you can observe in the blue measurements by these two lines. So that's like full inspiration and full expiration. And we see a lot less of that if we use a mechanical epicardial sensor. So in summary, we have approached the problem of post-cardiac surgical complication management, in particular the risk of vasoplegia. We have come up with a mechanical sensor solution that is implanted on the epicardium after surgery. So in conclusion, we showed that implantation of a soft strain sensor alongside with other things that are already implanted on the epicardium, like a cardiac defibrillator, could help monitor heart health continuously in the future. So the second example of cure where volume is important is really going back to something that I've introduced at the beginning of this talk, and that's really the pump speed. So we have learned back then that a pump speed that is set too high can really reduce the volume that we are measuring inside of the heart. And if the pump speed is set too low, so for example, if a patient goes into exercise, then we can still have pulmonary congestion, even though the pump speed that was set in the clinic was really adequate. So if we want to respond to like these two cases of sleep on the left-hand side where our volume decreases and maybe exercise on the right-hand side when our volume increases, we would need to have a relationship integrated into these pumps that is similar to the healthy heart. So in the healthy heart, we find that the volume of the heart, so the end diastolic volume of the heart, is actually pretty linearly related with its work output. So currently, if we go into these different stages like sleep and exercise, we divert from this healthy heart function. So ideally, if we could measure volume in the heart continuously, we could give this as an input to ventricular assist devices to change their pump speed according to the hemodynamic state of the patient. So during my PhD, I really thought a lot about how we can do this without creating an additionally new, very complicated sensor system. And one thing that I came across was that the end diastolic volume, so the volume at the end of the cardiac cycle, so the maximum filling volume, is actually inversely related to the measured amplitude of the ECG. So the ECG is really just the electric depolarization of the heart. So we're using the electric activity of the heart itself to help us identify the end diastolic volume. So this effect is called the Brody effect, and it has been measured previously on the surface, in the surface ECG, so those electrodes that we typically get attached to when we're in the hospital. However, the problem with measuring on the surface is that we also measure changes in position of the heart at the same time. This is why we conducted an in vivo study where we looked at if we could measure it using electrodes that are placed in the blood pool of the heart. So this worked well. However, it still required an external reference signal, which is why we then developed a novel unipolar electric circuit that would allow us to measure unipolar electric depolarization of the heart from the cannula of a VAD. So we then integrated just normal pacemaker electrodes into a dummy 3D printed and biocompatible cannula and implanted this in 6 animals. Again, we performed the same hemodynamic perturbations. So first, we looked at large and small changes in intravascular volume, where we really aimed at achieving a stationary point. And then secondly, we looked if we could use the calibration based on those stationary points to really predict into the future what would happen during an acute occlusion and an acute increase in ventricular volumes. So the first thing that we showed is that our intracardiac signals are reproducible across leads. So we first of all, obviously also have the surface electrocardiogram, but then also the lower plot, which shows us the intracardiac measurements, really showed very nice signals. When we now look at the sensitivity and the limits of agreement during those steady state calibration experiments, we see that in all, I'm showing only 4 animals here, but that in all 6 animals, we have a significant correlation between the maximum filling volume and the amplitude of the intracardiac electromyogram. And also we had a pretty good limit of agreement of about 10 milliliters overall animals. So in the second step, we then showed that during an acute increase of volume induced by an occlusion of the aorta, we first of all see that increase in volume on our reference measurement in black. And you can already notice in just the ECG measurement that we definitely have a reduction in amplitude measured by our unipolar amplifier. So if we use this to predict volume, we see that we definitely have a dynamic response of the measurement of amplitude, which is pretty similar to what we're measuring using electric impedance. So in summary, we have addressed the problem of physiological control of heart pumps to avoid extreme volume states. We have come up with a solution that is based on the intracardiac electromyogram. So in conclusion, it is definitely possible to continuously monitor volume from the cannula of a ventricular assist device just using off-the-shelf electrodes. So we have seen a couple of examples on how sensors can predict, prevent, and cure cardiovascular disease today. All of those applications have mainly focused on the systolic heart failure, so really the problem of the heart to contract enough. However, we also now know that 50% of hospitalizations are actually caused by a very similar but still different disease, which is called diastolic heart failure, where the problem no longer is the ejection of blood, but actually the filling of the heart to a maximum volume that improves ejection. So this is really one of the areas that I think we as engineers can contribute a lot by developing new systems, in particular because unfortunately for those patients, there are still very limited treatment options. There are very heterogeneous cohorts, so it's really hard to diagnose those patients for heart failure. And in some ways that's of course sad, but I think in other ways it's really helpful because now we can, as engineers, engage at the very start of developing therapies for a certain patient population where there is little standards already in place that might make it more difficult to adapt such technologies in the future. So where do we go from here? I hope that this conceptualizing of sensors together with different action takers in this control system perspective has maybe inspired some of you to think about in which other situations could we include an automated feedback for the clinician, for the device, or for the patient. And I've just listed a couple of examples here, listing from clinical decision support systems to predicting the interaction of devices and patients, or potentially automating the administration of drugs. A little bit further then to the patient, where we really give the patient some sort of tool to engage with their health in an automated way, but directly. The last thing I would like to touch on is the relationship between our research visions and findings and their counterpart in society. So a friend of mine works at the German healthcare policy, and she pointed me towards this position paper that describes the key areas in which they want to invest in. And it was just so beautiful to see how those key areas really align very well with the goals that we have set ourselves at Stanford Medicine and also in the PHIND cohort. So clinical decision support system, digital medication management, and then of course also robotic systems and telemedicine. So with this, I would just like to thank you, Stanford, for giving me the opportunity to engage with you all. I would like to thank Dov McElhinney from Cardiac Surgery for bringing me on originally as a translational fellow to work at the interface on engineering and cardiovascular applications. Then Daniel Ennis, who's been an amazing mentor over the last 2.5 years. He has a wonderful group that I'm showing here as well that really works together to create the best science that we can. Thank you to Alison Marston for her input from the computational side. I've learned so much from you and your group already. And then also I'd like Ellen Roach, who is co-mentoring me right now on the soft robotic projects that I haven't talked about a lot today, but that I will hopefully be able to talk about in the future. And then thank you so much to all my funding bodies, the NIH, for the Veteran Health Administration to help let me use their MRI scanner, and then my personal funding from the Swiss National Science Foundation. Thank you very much for the opportunity to talk today.
Great. Well, thanks so much for speaking at the PHIND Seminar series, and for a very excellent talk. I really liked all the different examples of how you're applying your work to these different contexts. For questions for our speaker, please go ahead and you either write them in the Q&A or raise your hand and we can call on you. And I guess while folks are coming up with their questions, I had a question. So you had one of your last slides was kind of showing the kind of the patient, the physician. Generally those loops kind of, a lot of times the patient likes to be involved and maybe they shouldn't be, or vice versa. It gets complicated. So I guess, coming from your perspective, how do we integrate the patient and the physician with this data so that we don't create alert fatigue, but then maybe they feel like they're still being able to look at their data? What do you find through your work?
So I think what I find through my work is that it's a question that is under asked, because usually there is a certain medical problem and then we talk with clinicians and then we find a solution, but often the patient is not involved in defining these research questions. So I think, especially also, for example, with physiological control, right? We can definitely build a system that automatically adjusts the pump speed to what we're measuring, but that might create very uncomfortable circumstances for the patients at some point. So maybe they would actually prefer to just have 3 stages for sleeping, relaxed body positioning and exercise that they could go into, rather than relying on a technology that might affect their perception and their well-being. So I think for me as a researcher, it would be wonderful if there was more engagement between not only just me interacting with or using patient data to make better products, but also to at a very early stage of these projects, engage with patients and ask them what they would really like to be better in their medical therapies.
Yeah, absolutely. I think a lot of larger studies, especially, seem to really pull in these different patient or public engagement groups that can add insights into this, and that makes a lot of sense and could certainly help kind of balance these things out, because certainly it's not an easy answer, I think, beyond the technical aspects. I guess another question I had was just out of interest, what are you going to be working on in your new position? And that's really exciting. You're starting so soon, and it sounds really great. Certainly along this flavor of items, but it'd be great to hear about, what new projects will you be championing when you go to your new position?
Yeah, thank you for that question. I think one of them I talked on, at least or Gary talked on in the introduction. So I am working a lot in the soft robotic space right now. So I think one thing that still I struggle with those ventricular assist devices we have right now is just how invasive they are, how they cannot be turned off, how they are in contact with blood, which can cause stroke, which would leave a patient in an even worse position than they were before. So I'm working on a soft robotic device that would wrap around a blood vessel from the outside. So you basically are adding a pump without directly engaging with blood. So that's one thing that I'm working on right now. And I really hope that such devices could potentially also be useful in pressure-induced diseases such as diastolic heart failure, as I briefly touched on. So that's definitely one area that I'm going to engage with, and ideally that would also be integrated with sensors to understand how it interacts with the physiology of the patient. Then the second thing that I'm really interested, that I got really interested in, is the interaction of our cardiovascular system and movement. So especially during running, there is a rhythm to running that we have. There's a rhythm to breathing that we have. And there's obviously also a rhythm to the heart. And I think that's just an area that is still very loosely understood. So how can we use movement to help the heart, to help breathing, to help movement? So just, yeah, how can we help patients be more effective in doing exercise and engaging with their heart potentially as well?
Excellent. And I guess, where do you see the field going when you're looking at kind of the, the prevention side, especially when you're talking about this movement? A lot of people have, different types of fitness watches or different types of sensors. I mean, a lot of these things are to be determined. There's signal processing issues. I mean, you know this very well. What could we be doing better? What do you think, where are we going in this aspect of being able to capture things before we get to the opportunistic of, okay, you have an implant, and I'm going to leverage that now with the tools that you're developing?
I think the first thing we need to do there is just to make sure that we're measuring the right things. So we're measuring step counts now for a while, and I think those have shown to be very motivational for people, especially just to bring more movement into their everyday lives. So I think that's one very impressive example where we already put the person in the loop and gave them an option to feedback their own system into a more healthy state. So that, I think, is really kind of what motivates everybody in this space. I think in terms of measuring ECG, we're still at the very beginning. So ECG is one of the most easy parameters to measure from the outside. Right now, we still need to touch the thing on the Apple Watch, right, to get a good ECG. I remain skeptical of heart rate monitors based on those wearable devices that are just on my wrist, just because, again, we use them during exercise and we don't know if what we're measuring is actually the movement or if it's the heart, or so those things I think we need to do more research on. But I definitely think there's a motivational aspect to these wearables in terms of getting people engaged with exercise more, integrated into their daily life, which I think is really the most realistic way in which we can improve exercise, because there's just so many things that get us distracted during our everyday chores, and I don't think like forcing people to exercise is gonna be a very long-lasting effect potentially.
Sure, absolutely. I've been hogging the airwaves here. Any questions from our speaker? Feel free to raise your hand or write something in the chat, in the Q&A, I should say. I was just pausing to give people a chance.
Sure. That's good.
I guess one question I also had was kind of while we're waiting is maybe, how long do those implants last when they're in, and what could be from the engineering side, what would you like to see improved, I guess, to be able to sustain or give more data, or whatever you think is the critical, the tipping point for these?
Probably two things. The first thing is they don't have any sensors, so I would add sensors. The tricky part with adding sensors is that it throws back the entire development process to the starting point. So we're looking at 15 years from like having a final design that has all the sensors working on the bench to having this implanted in patients and having them profit from it. So yeah, I would love to see sensors. terms of mechanical stability, I think the devices that we have are super robust. Again, the problem is that they're super robust, but also super sturdy and stable, and just not conforming to anything soft that we have in our body. So I think I would there just suggest to look into all the things that people have come up with in the field of soft robotics, and trying to understand how we can use more soft materials. However, then I think we will probably have to go further to the material scientists, because as soon as we talk about soft materials, the durability really becomes an issue pretty quickly. So I think the systems that are there right now work well. They're engineered to last, but we need something that is soft, which is going to be harder to make durable.
Absolutely. And how about the powering of the devices? Do you find that to be sufficient? Or I know, there's a lot of different ways people do it. Sometimes they use like ultrasound or different ways, but maybe not be as practical for other reasons. How do you feel about that aspect of the devices?
Well, so I think the ultrasound that might be for pacemakers. So so pacemakers, the only thing they do is to give an electric pulse every once in a while. So that really doesn't require so much energy. So we can really have these batteries that are implanted and are exchanged. I don't want to say something wrong here, but about every 5 or 8 years. With ventricular assist devices, it's a lot more difficult, because as they are supporting the pumping function, the power that they need is much more than a battery could give them. So what you really are looking at is a cable that comes out of the body and is attached to a battery that you carry on your waist. So I think that's another, like, besides stroke, that's one of infections are one of the most dangerous part of implanting these devices, which is why people really are starting or have started to look into transcutaneous energy transfer. So that's, if this is your chest wall, then you had a plate on this side and a plate on the inner side, and then you would no longer have a kind of a drive line go through the chest wall anymore. So I think that's going to be a big life-changing element once that becomes available.
I see. Yeah, that makes a lot of sense. Yeah, because these devices work a lot harder than some of the other ones you mentioned. So.
They do, yes, yes, absolutely, they do.
All right. Any questions for our speaker? All right. Well, it's been really excellent talking to you and learning about what you've been doing, and congrats again for your future position, and really appreciate all the insights you shared today. So thanks so much for being part of this.
Thank you very much, Ryan. I appreciate the opportunity, and yes, I will see you back hopefully.
Yeah, we certainly hope so.
Absolutely. Take care.
All right, everyone. Have a great day.
Bye, everyone.